Hello everybody, today we're going to talk about transcription. This is the first of two videos on transcription. So why is this relevant for the USMLE Step 1? Well, transcription is a commonly tested USMLE Step 1 topic because various diseases are associated with disruptions in this mechanism. An understanding of the transcription process and its associated terminology is essential for understanding common question stems as well as related USMLE topics. This can be an overwhelming topic because it is extremely broad and has some confusing terminology. Fortunately, you only need to understand the basics of transcription for the USMLE Step 1. By the end of this video, you will have a clear understanding of the most important elements of transcription, and then in part 2, we will go over the steps of transcription. So with that said, let's get started. So as you probably already know, transcription is the process of making RNA from a DNA template. Not all RNA made during transcription is used to make DNA. RNA that is not used to make proteins is called non-coding RNA. We will talk about non-coding RNAs later in this lecture. Transcription is essential for life, and errors in transcription can lead to disease states, or even death. Transcription is an extremely complex process and an area of intense study. Fortunately, you only need to be familiar with certain elements of transcription for the purpose of Step 1. We're going to cover each one of these elements in depth. Specifically, you need to be familiar with the following elements, that is, their role and function. These are regulatory sequences, transcription factors, promoters, RNA polymerase, post-transcriptional modifications, and types of RNA. So let's talk about the first of these, which is regulatory sequences. Regulatory sequences are special DNA sequences which transcription factors bind to in order to influence how a gene is transcribed. Regulatory sequences are named based on their effect on gene expression. So for example, a silencer binds transcription factors called repressors. These transcription factors typically decrease transcription. An enhancer binds transcription factors called activators. These transcription factors typically increase transcription. Unlike promoters, regulatory sequences do not necessarily need to be located upstream to the gene. They can be located anywhere within the chromosome. So generally speaking, there are two types of regulatory sequences: silencers and enhancers. Now let's talk about transcription factors. Transcription factors are proteins which bind regulatory sequences in order to regulate gene expression. Transcription factors are either activators or repressors based on their effect on gene expression. Activators bind regulatory sequences called enhancers and increase transcription. Repressors, on the other hand, bind regulatory sequences called silencers and decrease transcription. So just like how there are two types of regulatory sequences, there are two main types of transcription factors, that is, activators and repressors. Activators bind regulatory sequences called enhancers, and repressors bind regulatory sequences called silencers. Now let's talk about promoters. Promoters are DNA sequences where RNA polymerase binds in order to initiate transcription. However, transcription factors can also bind promoters in order to alter gene expression. Typically, a transcription factor will bind a promoter in order to block access to RNA polymerase and inhibit transcription. This is a very common mechanism. Promoters typically contain special DNA sequences called Tata boxes and Cat boxes. Essentially, these are long sequences of repeating thymine and adenine nucleotides in the case of a Tata box, or repeating cytosine, adenine, and thymine nucleotides in case of a Cat box. What you need to know is that a promoter contains repeating TATA sequences or CAAT sequences. Unlike regulatory sequences, promoters are always upstream to the gene being expressed. That is, they are located before the gene when considering the orientation of RNA transcription. The next thing you must be familiar with are the different RNA polymerases. RNA polymerases are enzymes which synthesize RNA polymers. Prokaryotes have one type of RNA polymerase which makes all the RNA in the prokaryotic organism, while eukaryotes have three types of RNA polymerase which make different kinds of RNA polymers. They are labeled RNA polymerase 1 through 3. We're going to talk about each one of these. So as we said in the previous slides, eukaryotes have 3 types of RNA polymerase labeled 1 through 3. Let's start with RNA polymerase 1. RNA polymerase 1 makes ribosomal RNA, which is the type of RNA found in ribosomes. RNA polymerase 2, on the other hand, makes messenger RNA and another type of RNA called small nuclear RNA, or snRNA. snRNA is a non-coding RNA. We're going to talk about non-coding RNAs at the end of this lecture. RNA polymerase 3 makes tRNA, or transfer RNA, and it also makes another type of ribosomal RNA. Lastly, we have prokaryotic RNA polymerase, and as we said before, this is the only type of RNA polymerase in a prokaryote. This means that prokaryotic RNA polymerase can make all of the different kinds of RNA that we've described, that is, ribosomal RNA, messenger RNA, and tRNA. So probably the most important RNA polymerase to know is RNA polymerase type 2, because this is the type of RNA polymerase which makes messenger RNA. So let's move on to the next important element regarding transcription: post-transcriptional modifications. Generally, there are 3 mechanisms involved in post-transcriptional modifications. They are removal of introns, which involves a spliceosome, addition of a 5' cap, and addition of a poly A tail. Removal of introns requires the use of various proteins and ribozymes, which together are known as the spliceosome. Ribozymes are RNA molecules which function as enzymes. The non-coding RNA molecules known as snRNAs are also an important component of the spliceosome. We will talk more regarding snRNAs and other non-coding RNAs at the end of this lecture. The 5' cap of mRNA contains high concentrations of modified guanine nucleotides. It functions primarily to stabilize mRNA, help mRNA bind to the ribosome during translation, and prevent the degradation of mRNA. The 3' cap of mRNA contains high concentrations of adenosine nucleotides, and it functions to prevent mRNA from being degraded. So what I want you to know regarding the 5' cap is that it protects mRNA from degradation, and it also helps bind the mRNA to the ribosome during translation. And what I want you to know regarding the poly A tail is that it's very important in preventing degradation of the mRNA. If you remove the poly A tail from an mRNA molecule, it will be rapidly degraded and it will not be translated into protein. Another important characteristic of transcription that I want to introduce you to is called alternative splicing. Now, most proteins are made from mRNA transcripts which contain a sequence of exons. By changing the order of exons or removing certain exons, different proteins can be made from a single gene product. So for example, the gene on the illustration could produce 3 different proteins based on how the hnRNA or immature RNA is spliced. So for example, let's say that you have a gene in DNA that produces this type of hnRNA or this immature mRNA. Now, you could theoretically make various kinds of proteins from this single RNA molecule by rearranging the exons. So for example, you could remove one of the exons and you'll produce one protein, or you could switch the order of exons and you'll produce a different protein. This is alternative splicing. This mechanism explains why it is believed that more types of protein exist than there are genes present in the human genome. So, as we mentioned before, not all RNA functions to make proteins. RNA that is not used to make proteins is called non-coding RNA. Dozens of non-coding RNAs have been described. However, you only need to be familiar with 5. Let's start with MicroRNAs. MicroRNAs bind mRNA, block translation, and increase mRNA degradation. Therefore, they function to decrease translation. We're going to take a closer look at microRNAs in the next slide. Small interfering or siRNAs work similarly to microRNAs. However, they stop translation by actually cleaving the mRNA molecules, which is something that microRNAs do not do. We're going to look at these a little bit closer on the next slide as well. Short nuclear RNA, or snRNA, forms a part of the spliceosome and plays an essential role in RNA splicing. We already mentioned this. rRNA, or ribosomal RNA, forms parts of ribosomes, which are very important for the production of proteins. tRNA, or transfer RNA, is essential for translation and synthesis of proteins during translation. They function as the amino acid carriers. So now let's take a deeper look at microRNAs, siRNAs, and how they work. So here we have a simple illustration highlighting the steps involved in microRNA and siRNA function. As we mentioned before, both of these mechanisms work to inhibit translation of protein. The steps involved in both are very similar in that they both involve formation of an RNA hairpin. First, an RNA molecule is synthesized by RNA polymerase, which forms a hairpin structure. This is because both ends of the molecule have complementary base pairs. So for example, we can see a hairpin looking structure on both of these systems. Second, an enzyme called dicer cuts the RNA in order to form a double-stranded RNA molecule. So you can imagine that the head of the hairpin is cut off by this protein or enzyme called dicer. So this is where the paths diverge. In the case of microRNA, one of the RNA strands is removed and the other strand binds a complementary sequence on an mRNA molecule. This microRNA molecule then obstructs translation and decreases the time required for the mRNA molecule to be degraded. So we can imagine that this microRNA will bind a complementary sequence on this red mRNA and will obstruct the ribosome and the entire process of translation. Another effect of microRNAs is that they increase the rate of mRNA degradation. So in a nutshell, a microRNA will stop the process of translation and increase the process of mRNA degradation. Now let's look at siRNAs. In the case of siRNA, one of the RNA strands binds a complementary sequence on an mRNA molecule similarly to how microRNAs do it. However, instead of physically blocking translation, siRNA will recruit proteins which will cleave the mRNA molecule. These proteins that are recruited by siRNA together are called the RISC, which stands for RNA-induced silencing complex. These proteins will cleave the mRNA, and again, there will be no translation, no protein production. You do not need to be familiar with all the steps involved in the microRNA and siRNA system. What you need to know is that both mechanisms work to decrease mRNA and translation of proteins. That is, they stop translation. Lastly, we're going to talk a little bit about P-bodies. P-bodies act as management centers for mRNA and non-coding RNA. P-bodies are believed to be involved in post-transcriptional regulation of gene expression. That is, they are another mechanism which can influence protein production by controlling the availability of mRNAs and non-coding RNAs, which, as we said before, can influence translation. The role and function of P-bodies is still debated, but what I want you to know about P-bodies is that they are collections of proteins, mRNA, and non-coding RNAs which can influence translation and protein production. Specifically, they can do this by degrading mRNA, storing mRNA, or storing microRNAs, which can degrade mRNA and stop translation. So in summary, transcription is an extremely complex process which involves the action of various enzymes, proteins, and RNAs. What you need to be familiar with regarding transcription are the function of regulatory sequences, transcription factors, promoters, RNA polymerase, non-coding RNAs, and post-transcriptional modifications. If you have a basic understanding of these elements, you're ready to understand the specific steps of transcription, which will be the topic of our next lecture. Thank you for watching and see you on the next lecture.